Okay, ladies and gentlemen, you are welcome and good afternoon, all. Can you hear me? Hello, 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 okay, Chika Ejim, you are welcome. So, I think if you could remember, ladies and gentlemen, we have been having a lot of network issues or network problem concerning our classes. So that is why I think this time around we are going to have live class this time around so that at least will not have embarrassment from the network. So I think if you look at it from the introduction or from the title, we are actually going to look at the concept of cell biology because we have discussed or we have done a lot of things on the concept of cell biology. So that is why we are going to recap them. And I learned that so many of you are not even watching our videos. So when we send some of you videos, we don't used to have time to watch. So please try to endeavor and watch our videos because most of those things that you are seeing are the things that can actually come in my examination. So now, do you have any question concerning our lectures, our previous lectures, please? Do you have any problem? Do you have any question? Do you have any questions? Do you have any question? Okay, let me just actually recap. So I just want to tell you that I think we look at the okay, let me actually give you a brief introduction of the journey of life because that is where we're supposed to start last time and we have a lot of network difficulties. So I actually in the journey of life, when we said journey of life, it means how the life started. And of course we look at how life started from atoms, how atoms form molecules up to the point of a cell. So now, how life started, it started from sperm and egg. So last time I told you the largest cells that is found in humans, the largest cells is the egg cell. And the smallest cells that is found in humans is the sperm cell. So what usually happened, ladies and gentlemen, during fertilization? Egg and sperm cells come together. And I even give you the real definition of fertilization, where I told you that fertilization is not just a combination or a union of egg and the sperm cell, where I told you that actually fertilization it is actually associated with the union of the nucleus of the sperm cell and the union of that of the egg cells. That is how fertilization starts. Please, I have 1 minute. So actually, what happened is actually when a sperm and the egg cells, the nucleus unite. And I think the last time I sent you a picture on the structure of egg and sperm cell, let me also show you the structure of sperm. Structure of sperm sperm cell, so that at least you can look at it very well and enjoy it. I think if you could remember the last time I told you that Excel is has three major components. It has three major components. And these three major components include head, neck, and tail. So let me show you so that you can clearly see how it is. So that you can really get the concept very well. Let me show you. I hope you can see this. Please, can you see this? Can you see this picture, please? Okay. So you see, the three major components of egg include head, and then it includes the neck, and then the tail. So if you look at it in the head, in the head, we have nucleus because look at it. If you look at this neck, the head contain three components. We have acrosome, the cycle, the cycle in maroon is the nucleus. And then we have centriole. And then in the middle phase, we have mitochondria. And then the tail contain flagellum. So what is the importance of the mitochondria? Remember, ladies and gentlemen, during sexual intercourse, man need a lot of food because one of the things that actually demonstrate the quality of the sperm is it is motility. How the sperm was able to move from where it released to where it's going to meet the egg for the fertilization. So it needs a lot of energy. And the energy, remember, is actually coming from mitochondria. So in mitochondria, if you could remember from the concept of the cell, the major function of the mitochondria is the powerhouse of the cells. It generates most of the energy. Most of the energy is coming from mitochondria. So it is the one that gives the sperm cell energy. And the tail, if you look at it, the tail contains flagellum. And the major functions of flagellum is the motility, that is the movement of the sperm cell. Please, do you understand this? So we have acrosome. In the acrosome of the egg, we have what we call an enzymes, and the enzymes is called hyaluronidase. So usually sperm cell releases an enzyme called hyaluronidase, and the major functions of this hyaluronidase is to break down the nucleus, so it's to break down the zona pellucida of the egg so that the nucleus can easily be released for the fertilization to take place. So I want you to understand, ladies and gentlemen, in the entire of this sperm cell, it is only the nucleus of the sperm cell that can get into the egg for the fertilization to take place. And that is what I want you to understand. Some of the egg are infertile because they cannot break down the hyaluronic acid that are found in the zona pellucida of the egg for the fertilization to take place. So that is what I want you to understand. So, and very importantly, after fertilization, we have a zygote, and a zygote is a single cell which contain a DNA. So the sperm cell and the egg cells, when their nucleus come up together, there is a formation of a zygote. And how this zygote is formed is as a result of the combination of the nucleus of the sperm cell and the nucleus of the egg cells. And as a result of that, there will be a genetics recombination where you have a single DNA as a so a DNA in a zygote is like a recombinant DNA. Why do we call it recombinant DNA? Is because it comprises the DNA from the mother and the DNA from the father. And this is the reason why you look at your child, you will realize that some characters are coming from the father and some characters are coming from the mother. It's because of this genetics recombination. During fertilization, meiosis takes place. So that is why I want you to understand this very important. Meiosis take place during fertilization. So during this fertilization, the meiosis take place. Therefore, you are going to have a recombinant DNA, and this recombinant DNA is as a result of the combination of DNA from the mother and from the father, and that is why our child look look like their father and mother. So we have a maternal character from the father and maternal character from the mother. So that is what I want you to understand. So after that, a zygote is a single DNA, and that single DNA will, from that single DNA in the zygote will divide into two, three, four, up to 16 to produce what we call a morula. So I want to also give you something here very important. Morula and the zygote, from there you can harvest what you call stem cell. Morula and the zygote, the cells that are there, because after there is what you call a cleavage. From zygote, cells will cleave to divide from 1 to 2, 2 to 4, 4 to 8. So mitosis will take place. So meiosis during fertilization and mitosis during cellular division, where a zygote cells will divide into 2, from 2 into 4, from 4 into 18. Sorry, from 4 into 8, from 8 into 16. And this a cell during development that has 18, sorry, that has 16 to 32 cells are called morula. And from there you can harvest stem cell. So now let me ask you, ladies and gentlemen, what are stem cells? Who can define stem cell? Who can define stem cell? Who can define stem cell? I know that some of you that watch my video on fertilization in anatomy, you can actually define that. What is fertilization, please? What is fertilization? What is fertilization? Sorry, what are stem cells? I'm talking about stem cells. What are stem cells? So I think let me just define. Stem cells are the cells that have the potential to differentiate into cell types. So stem cells are the cells that have the ability. So stem cells are the cells that have the ability to differentiate into different cell types. From stem cells, you can produce eye cells. From stem cells, you can produce kidney cells. From stem cells, we can produce pancreatic cells. From stem cells, we can produce brain cells. So that is what we call stem cells. You see, someone is still giving me the same definition. Fertilization is a process by which the male sperm cell and the female cell join to form a zygote. This is wrong and it is not correct. So stem cells are the cells that have the potential to produce different cell types. From these stem cells, we can produce eye cells. From these stem cells, you can produce brain cells. From these stem cells, you can produce kidney cells, and that is what we call stem cells. And we have different types of stem cells. Please, I hope you are jotting. Number one, we have totipotent stem cell. We have pluripotent stem cell. We have multipotent stem cell. And then number four, we have unipotent stem cell. So these are the different types of stem cells based on their differentiation ability. Because stem cells are classified on different bases. So based on their differentiation ability, the word totipotent stem cells, it means they have the ability to differentiate into all cell types. All cell types. Remember, I told you in human we have how many different cell types? How many different cell types do we have in average human being? We have over 220 different cell types in human. So it means the totipotent stem cell, it means they have the total potential to differentiate into 220 different cell types. Excellent, Aisha Mukhtar. And we have pluripotent. So pluripotent stem cells, they have the ability to differentiate into some cell types. So they have limitation. They are not actually be able to differentiate into all cell types. So they have some limitation. There are some cells that cannot be generated from pluripotent stem cells. Please, I'm giving you this. I want you to understand something. There is a difference between the video that I used to send you and also the explanation that we usually have in the live session. Because the importance of the live session is for you to have a good understanding of what the biochemistry is all about, what message that is actually trying to give in the aspect of medical science. Because all this thing, the issue of cellular differentiation and specialization is as a result of the DNA from the zygote. So that's what I want you to understand. So when you have one cells, that is zygote cell, containing one DNA, it will give you a cells that can differentiate into all cell types. And how is that happening? It's because of the DNA. Remember the last time I told you in the DNA we have about 25,000 genes. And these 25,000 genes, they have the ability to differentiate into or to express a particular proteins to make one cell specific from one another. Like for example, now let's get it. We have a zygote containing DNA differentiate into, let's say, the zygote cell, the same zygote cell that contain DNA, and that zygote cells will divide into millions of cells. And remember, in mitosis, when one cell differentiate, the daughter cell and the mother cells and the parent cells are 100% identical. The zygotes, the daughter cells and the parent cells are 100% identical. They contain the same DNA, They contain the same genetic material or the same kind of mitochondria. So you have one cell differentiates, so the one cell, which is zygote cell, divides itself into, let's say, 1,000 times. So what happens after you have, let's say, 1,000 cells? So some of those 1,000 cells originated from single zygote. They will actually, some portion of that 1,000 cells will be allocated, and those allocated cells will now instruct the DNA to produce a particular protein. Like, for example, you have 1,000 zygote cells which originated from single zygote through mitosis. So some of the cells will be allocated, and the DNA there will be instructed to produce hemoglobin. So once hemoglobin is produced from those allocated cells, they become red blood cells. Some cells will be allocated, and then the DNA there will be instructed to produce brain cells. So therefore, the neurotransmitter will be expressed from that DNA, and then those cells are now specialized as neurocytes. That is the nerve cells. Some cells will be allocated to produce or to express some proteins, like, for example, insulin, glucagon, somatotropin. So if some cells are allocated to produce glucagon, insulin, and somatotropin, that cells will be specialized as which types of cell? Please answer me this. That cells will be specialized as what type of cell? When you have some zygotic cells or zygote cells allocated, and the DNA there is instructed to produce glucagon, insulin, and somatotropin, which cells are going to be that? What are the cells? Those cells will be specialized into what, please? No, it's not hepatocytes. I'm talking about glucagon, insulin, somatotropin. Let's these three proteins. Let's who can give me the answer? What is the answer, please? It's not hepatocytes. Excellent, Aisha Mukhtar. They are pancreatic cells. Remember, even the pancreatic cells, we have different pancreatic cells. Like, for example, in the pancreas, we can have beta cells of Langerhans. That we have beta cells, we have epsilon cells, we have delta cells. All these are different cells that constitute to form pancreas as a tissue. And also at the same time, some zygote also generated from that single zygote will also be allocated to differentiate or to express some proteins which make them to produce liver cells. Then some cells will also be allocated and instructed to produce actin, myosin, yes, myosin, actinomycin, and actinomycin and myosin. So those cells will be differentiated into muscle cells. So that is what I want you to understand. So it means that the DNA is what gives different organs in our body, is what gives different tissues in our body. That is what I want you to understand. Liz, do you understand now how DNA is important in cell? So when we say cellular specialization and differentiation, it means how cells differentiate itself based on the genes or proteins that are produced in that specific cells. So that is what make one cells differ from one another. Please now, do you understand the concept of cellular specialization and how DNA play a critical role in cellular differentiation and cellular specialization? So what make one cells differ from one another, one organs differ from one another? The answer is differential gene expression. The DNA that is there in the kidney is the same thing with the kidney with the DNA in the brain. It's the same thing with the DNA in the liver. It's the same thing with the DNA in the brain cell. It's the same thing with the DNA that is found in the eyes. But what make one DNA from one another is because some genes are on in particular cells, while some genes are off in particular genes. So there is on and off of some genes. In the like, for example, in the brain cells, you have a gene that can produce insulin, glucagon, somatotropin. But the genes are there, they are silenced, they are silenced. While in the kidney, those insulin, glucagon, and somatotropin genes, they are activated, they are on in the pancreas. That is why glucagon, epinephrine, and insulin are produced. And also at the same time, in the pancreas, you have a genes that can produce neurotransmitters and some important enzymes in the brain, but they are off there. So this is what you call differential gene expression. So differential gene expression is what make one cells differ from one another. But the DNA that you have in your hair, the DNA that you have in your saliva, the DNA that you have in the kidney, the DNA in the brain, they are all the same. Please, is this point now clear? I'm just trying to do this so that for you to understand the DNA as a language of life. Because remember, we started discussing on the different organelles of the cells, where we talk about the nucleus, and of course we are coming to the mitochondria and also the endoplasmic reticulum. So I'm just doing this so that at least whenever we said DNA, as a student of DLC, you should be able to know what DNA is all about or what the nucleus is all about. The nucleus contains DNA, and the DNA is a language of life. Is what make you who you are. Is what make me who I am. Is what make each and every organism differ from one another. Human is not like a god. That is goats. It's not the same as cow. It's not the same as sheep. We are all different from one another. And what makes this different is the DNA. So that's what I want you to understand. Now let me ask you another question. Assuming now you take the sperm and you take the egg cell. So when you take the egg cell and you remove, here we have amino acid, we have Cecilia, Azuba Cecilia. So let's say now you take the egg of the Cecilia and then you take the egg of amino acid. Then you remove the nucleus of the egg cell from Cecilia, and then you now transfer the nucleus of the egg from amina into the egg of Cecilia. So you knock out the nucleus of the egg cell of Cecilia, and you now put the nucleus of the amino acid. And after you can call this actually recombine. For those of you that watch my video on cloning in Anatomy 209, we talk about cloning. We talk about reproductive cloning, and this is how it started. This is how cloning started. So you remove the egg. So you take the egg, you remove the nucleus of Cecilia egg, and you replace the nucleus of the cellular egg with that of amina. And then after you have that combined egg, you have sperm fertilize it. Who can tell me now the character of the child? Is it going to be that of Amina or Cecilia? That is the question. Answer it, please. Okay, someone said only for Amina. Someone said it's Amina and Cecilia. Okay. So the truth is that 80% of the character will come from that of Amina, or let me say 90%. And very few characters will come from that of Cecilia. The reason is because the mitochondria of Cecilia is also still there. And remember, mitochondria also contain DNA. So higher percentage of the character of the child will come from Amina. And very few will come from Cecilia because of the mitochondria. Remember, the mitochondria also has DNA. Is that clear now? Excellent. So, after this, what next? So, I want you now to understand that, ladies and gentlemen, do you understand the concept of the nucleus and the DNA? Although we are also coming to discuss the DNA itself. We are coming to talk about the DNA itself because we'll do nucleic acids. Please, is that clear so that we can proceed? Do you understand? Is that clear so that we can proceed? Okay, excellent, Joy. So now the next thing that we are going to look at is I know that we also did cell membrane. Cell membrane. What is the importance of cell membrane? So I will explain cell membrane together with the transport system. Okay. Obas, Sunab Obas, ask you a question. Sunab Obas, ask you a question. Anyone, if you have any question, please ask. I'm ready to answer the question because that's the importance of even this live class because I send you a lot of videos for you to watch. So if you have any question, do not hesitate to ask your question. I'm ready and I will answer it. Yes, ask your question. One side of Amina, one side CC. What do you mean? I didn't understand your question. Oh, so now Obas, ask your question. Anybody that has question, please ask. Let me. I'm coming 1 minute. Okay, that is when the egg was donated by another woman and the sperm cells was that of the husband. And the zygote be exactly that of the wife whose egg was not used. And class na ke na online class ne. That is when the egg was donated by another woman and the sperm was that of the husband. Will the nucleus be exactly that of the wife whose egg was not used? I don't understand, Stunab Obas. Please ask your question again. Let's ask your question again. I don't understand. So that is when the egg was donated by another woman and the sperm was that of the husband. Will the zygote be exactly that of the wife whose egg was? This your question is not clear actually. So what I was saying actually, ladies and gentlemen, is that let me just repeat. Actually, your question is not clear, Obas. So that is actually the concept of fertilization and how nucleus is important in cellular differentiation because of the DNA. And then the next thing that we are going to talk about is the cell membrane. So what is the importance of cell membrane in a cell? The importance of the cell membrane is to control what will get in and out of the cell. That is the most important thing of the man, of the. Of us. Okay, and also it please in the case of IVL, I mean in donor tissue, what happens to the zygote at the end? No, when you said in vitro fertilization is different from the concept I explained. In vitro fertilization is when actually you have infertile male or infertile female, you get the sperm of the man and then you get the egg from the female. And there are different ways in which you can get the sperm cell into the egg. You can use injection. We have call it intracytoplasmic injection, where you can inject the sperm into the egg for the fertilization to take place. Because sometimes one of the major problems that make a man or female infertile is because of the inability of the sperm cells to get itself into the egg. So in the in vitro fertilization, you can make it in such a way that you can inject the sperm cell into the egg and then the fertilization will take place and then you allow the zygote in vitro, that is in test tube or in the laboratory environment to divide itself until when it reaches to the stage of embryo. That is when the embryo will be implanted back into the mother or into a surrogate mother for the child to develop. So that is actually IVF in vitro fertilization. So it is different from what you have said. What Obas is trying to ask is that in the case of IVF, when the husband's sperm cell will be used to fertilize another woman egg, will the child have the donor character or the wife that carried the pregnancy? I think I just explained it. If you get the sperm from the mother, the child character will be exactly that of the mother egg donor. So the mother, that is the female or the woman that donate her egg, the character will exactly come from the father. So if the egg was that of the donor, after the procedure, will the zygote resemble the woman? Of course, it will resemble the woman. The woman that donate her egg, it will resemble her. Although sometimes I want you to understand, it depends if there is a surrogate mother because there are some internal factors that can affect the phenotypic character. For example, let's see you now took the embryo after you culture it in the lab, then you implant it back to the mother. So there are some internal factors that can also contribute to the phenotypic appearance of the child. Because I want you to understand that phenotypic character or a character usually comes as a result of combination of the environment and the DNA. So that's what I want you to understand. Please, is that clear before we move to the cell membrane? Is that clear? Is that clear, please? Obas, please, do you understand what you asked? Okay. Okay, so Balikisiti Muhammad Obas. Okay, so now the next thing is the cell membrane. So a cell membrane is one of the very important part of the cells where it control what will get in and out of the cells. So because we take drugs and drugs also we take drugs and we take food, we take different nutrients, we take air. So how air, how nutrient, how drugs get in and out of the cell, so it's actually depend on the cell membrane. So now I want to ask question, what are the major component of the cell membrane? What and what form the cell membrane of the cell? What are the major component of the cell membrane? What are the major component of the cell membrane? What is the major component of the cell membrane? I know that you watched the video, so I want you to tell me. What? Okay, carbohydrate one. I agree with you. Chiamaka. Chiamaka says phospholipid, yes, excellent. Then what again? Yeah, I like it. Abbas Dawasa, phospholipid, protein, and carbohydrates. Yes, yes, and glycoprotein, excellent. No hydrophobic. Is the you didn't. It's actually part of phospholipid comprise of hydrophilic head and hydrophobic tail. So I want to, and there is also cholesterol. Excellent. Kilo Mariam. So let me mention them. Number one, the cell membrane constitutes of carbohydrate, proteins, glycoproteins, cholesterol, and phospholipid. So these are some of the important components of the cell membrane. Excellent. So what are the types of proteins that are found on the cell membrane? The types of proteins that are found on the cell membrane. What are the types of proteins that are found on the cell membrane? What are the types of proteins that are found in the cell membrane? No, globular protein, no, no, no. That is actually a general types of protein, globular protein. Excellent, excellent, Akila Mariam. We have integral and peripheral protein. Integral and peripheral proteins, yes. Excellent, Matida Job. You mentioned all the components. So we have integral protein and peripheral proteins. These are the types of proteins that are found on the cell membrane. So please, who can tell me what are the importance of integral and peripheral proteins on the cell membrane? What do they do? What do they do? Yes, what do they do? What are the importance of the proteins? They serve as a carrier molecules. Excellent, excellent, Eliasu Dawasa. They serve as a carrier proteins because they are molecules, because they are large or they are charged, they cannot get into the cell membrane. So they need a carrier protein. There is protein that can carry them, that can take them into or out of the cell. So those proteins they can serve as a channel or a carrier. Get it, please. They can serve as a channel where the protein can get in. That is one. And they can serve as a carrier because there are some molecules that are charged and they cannot get into the cell. So the proteins will carry them, will pick them, and get them into the cell. So you see, these are the two functions of integral and peripheral proteins. They serve as a carrier molecules. They are carrier proteins. They carry some proteins, sorry, some molecules because there are molecules that cannot get themselves into the cell. So if that is the case, they cannot get into the cell. So they have to be transported. So a proteins will carry them into the cell. So now I want to ask another question. Excellent, facilitate movement of substance in and out of the cells as carriers or channel. I want you to add with that belief. Excellent. So now, after understanding the concept of integral and peripheral protein, now that is what they do. So, what is the difference between integral and peripheral protein? Even from the word integral and peripheral, integral and peripheral, I think you can differentiate them. Integral proteins, what do they do? What are the difference between integral and peripheral protein? Just their difference, please. Excellent, excellent, Matida Joff. Integral proteins are important in the transporting of substances. So sometimes they can even act as a receptors. Yeah, but I want you to like Chidima. I like this your answer. Chidima Ela. Integral proteins is within the cells, so they are fully integrated into the cell. While peripheral protein from the word peripheral, they are just partially or transiently attached themselves on the cell membrane, and they usually found outside the cell. Excellent. Integral proteins are fully embedded, yes, are fully embedded within the lipid bilayer, while peripheral proteins attach loosely to the membrane surface. Excellent, Naima Mukhtar, and God bless you. Naima, you are my daughter, so God bless you. Excellent. So I like some of your answers, please. Excellent. So this is actually the difference between the integral protein and peripheral protein. So now the next question now is that You say that these integral proteins and peripheral proteins, they help in the transport of substances. So who can tell me which types of transport system use a protein, use a protein as a carrier to transport molecules into the cells? Which types of transport system is that? Is it active transport? Is it primary active transport? Secondary active transport? Is it passive transport? Is it simple diffusion? Is it facilitated diffusion? Which one? Is it uniport? Is it sympot? Which one? Yeah, yeah. Active transport. In the case of active transport, they require protein channel. Excellent. The answer is facilitated diffusion. Active transport require ATP and protein channel, but facilitated diffusion require a protein carrier. So protein carrier, that is a cargo that can pick molecules and get them in or out of the cell is facilitated diffusion. While active transport require protein channel. So it means both active and facilitated diffusion. Liz, I'm happy the class is getting interesting. That means you are watching the videos. God bless you. So this, if we keep doing this kind of live classes, that is how you will be able to pass your examination. So keep watching all those videos. I'm very sure it will definitely help you. And I want you to understand something. I have two YouTube channels. There is Science Made Easy and there is Science Academy. So the main YouTube channel is the Science Academy by Mukhtar. That is where I have close to 700 videos. And all the physiology tutorial that I am doing, the physiology tutorial, human physiology 225 and human physiology 227, biostatistic and biochemistry are all there in the main YouTube channel. But this YouTube channel is mainly for anatomy. So that's why I'm telling you, if you want to have access to all my videos, including the anatomy, the physiology, the biostatistic, you need to register with that Science Academy YouTube channel, because all this will really help you and will give you an insight on how to write examination. In fact, I even have fast question paper. I solve fast question paper with some videos. So there are some videos that once you watch them, I assure you'll hardly have a problem with your examination and your quiz. I'm sure about that. Physiology, the two physiology I have already covered. I have fully covered them. So if you are not in my tutorial group, please try to join. And if you didn't register with the YouTube membership, if you are interested, it's not compulsory, because all biochemistry videos are free. But the four other courses, if you want to have access to them, so you register and I make the videos based on their course material. So now the drugs in the pharmaceutical industry. What is the nature of the drugs from pharmaceutical industry? Which type of transport system do you think drugs use to get itself into the cell? Which type of transport system is not active transport? If you say passive transport, we have different types of passive transport. We have simple diffusion, we have facilitated diffusion, we have osmosis. So which one? If you say passive transport, so which one? Is it simple diffusion? Is it facilitated diffusion? Or osmosis? Which one? The answer is simple diffusion. But why do you think it's simple diffusion? Why do you think it's simple diffusion? Why do you think it's simple diffusion?
ఇంకా kana da tsinawa kana da key stuff na tunan
So my question is what, okay, please can someone help me? What is the question that lead to this to simple diffusion? I asked a question that which types of transport system do you think that drugs use to get itself into the cell? They mentioned different types of diffusion, and I said the answer is simple diffusion. And I said, why do you think drugs use simple diffusion to get into the cell? That is the question. Chiamaka Joy. Ezekiel Nozi. Yeah. Yes, the answer is simple diffusion. But why is simple diffusion? Transport system that use proteins are facilitated diffusion. The active transport, okay? Yes, 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 ladies and gentlemen. Why do you think drugs use? Okay, so I want to tell you something, ladies and gentlemen, very important. Since you don't know the answer, yeah, of course, it doesn't require, it requires no career. Naturally, ladies and gentlemen, you see, we talk about the lipid bilayer. The lipid bilayer, so any substance that is lipid soluble can just use a simple diffusion to get itself into a cell. Just simple diffusion. So just use a simple diffusion to get itself into the cell. So any substance that is lipid soluble. So I want to tell you something. Most of the drugs that are synthesized or that are produced by pharmaceutical industries are lipophilic in nature. Lipophilic in nature. What do you mean by lipophilic? Lipophilic are substances that are soluble in lipid. Take paracetamol, put it inside water. Does it solubilize? It doesn't solubilize because it's not water soluble. Take paracetamol, put it in water and see. It will end up as a suspension because it's not water soluble. So most of the drugs that are synthesized by pharmaceutical industry are lipophilic in nature. So because drugs are lipophilic in nature, so when we take them, remember where is the site of the drugs metabolism in a cell? Where drugs metabolism takes place in a cell? where drugs metabolism takes place in a cell? That is my next question. Yes, I know drugs is metabolized in the liver. So, but where specifically in the liver? Where? Excellent, Abbas Dawasa. You are always in point in the smooth endoplasmic reticulum. So, drugs are metabolized in the smooth endoplasmic reticulum. So drugs are metabolized in the smooth endoplasmic reticulum. So when we take drugs, the drugs will reach liver. So when the drugs it reach liver from the liver cells, then it will now move down to the hepatocyte, that is the cells of the liver. And then the cells of the liver will now, or the drugs will now get itself into the cells in order to reach smooth endoplasmic reticulum for the drugs to be metabolized. So pharmaceutical industries synthesize drugs as lipophilic so that, remember, as a patient, you always need drugs to be metabolized so that it can mitigate the disease condition. So that is why the drugs are generally metabolized in the form of lipophilic. If drugs are not lipophilic, maybe at the end they may end up abusing active transport. So active transport use a lot of energy before it transports substance into the cell. And most of the patients they are not taking enough food to generate energy. So if you are giving patients somebody that cannot generate energy or that doesn't have enough energy because they haven't eat, so what do you think? What do you think? It will end up the drugs not the drugs will be in vain because it is not going to do anything. Please, do you understand? Do you understand? The drugs cannot do anything at the end. If it is actually not lipophilic, it might end up abusing facility, it might end up abusing active transport, and active transport require energy. And if active transport require energy, so what will happen? The drugs might end up of doing nothing in the body. So that is why most of the drugs are lipophilic in nature. So once you take them instantly, that is why we even have different route of the drugs administration, depend on the disease condition. We have intravenous, intradermal, intramuscular, we have lingual, intralingual, we have so many of them. So because you need the drugs to easily get into smooth endoplasmic reticulum for it to be metabolized. So that is why drugs are synthesized as a lipophilic in nature so that it can easily cross the membrane of the cells to reach smooth endoplasmic reticulum. That is why it uses a just simple diffusion. Please, is that clear? And of course, remember, another thing that I want you to understand in simple diffusion is the types of diffusion in which a molecules move from the region of higher concentration to the region of lower concentration. Is that clear? Is that clear? Please, do you understand? So now I think number one, you understand the nature of the drugs that are produced by pharmaceutical industry. Most of the drugs they are lipophilic in nature. That is one. And second, why is it that they are lipophilic in nature? Because drug needs to be easily get into smooth endoplasmic reticulum for it to work, for it to metabolize and utilize to mitigate disease condition. Why is it that it is not in the why not the drugs are not water soluble because at the end it might require energy, and most of the patients are not taking enough food for them to get enough energy that can be used to transport the drugs. Does it make sense? Does it make sense? After this lecture, I will send you like just 6 minute videos. It's 6 minute videos to understand the different types of the transport system and their types, as well as the differences between primary and active transport. I will send you just a 6 minute video, I think. It's not more than 6 minutes. It will just help you. The importance I sometimes I use to do short videos, and the importance of doing short videos is to help you to easily grab the concept of whatever we are doing. Okay. So then, after this, what next? Is primary and so we understand that active transport require energy. Passive transport doesn't require energy. And active transport is divided into two. We have primary transport, we have active transport. We have primary active transport, we have secondary active transport. And then we have bulk transport system. Bulk transport system is divided into endocytosis, exocytosis, and pinocytosis. Do you need what? Adine, do you need what? Is it okay? Adeline, do you need what? What do you mean? Please, what do you mean? Do you need what? Smooth endoplasmic reticulum is where the drugs metabolism takes place. Okay, so now, yeah, of course you need it. You need them. So after this, um, okay, thank you, Bakar Ishaq Lawal. So now, the next thing after this concept of diffusion is so we have simple diffusion, we have facilitated diffusion, we have osmosis. And I know that you all know their differences. Facilitated diffusion, we explained it, it need a protein carrier. So the only thing that I want you to understand, because sometimes they ask this, understand each types of molecules that can be transported. This is part of the question paper that they usually ask. Like active transport is a types of transport system that require energy to transport molecules. And this active transport is divided into two. We have primary active transport, and we have secondary active transport. So let me ask, primary active transport require what? Primary active transport require ATP to transport molecules. It requires ATP to transport molecules. Excellent, excellent, Chika. Chantal Chika. Then after that, we have secondary active transport. So let me ask, secondary active transport, what do they require? No, vesically mediated is not active transport. Vesicle mediated transport is bulk transport. Bulk transport. So now the question is, secondary active transport require what? No, secondary active transport, it's not actually the major things required because we are talking about energy. Yes, vesicle mediated transport is the endocytosis and exocytosis, and that is not what we are talking about, Chiamaka. We are talking about the secondary active transport. The secondary Subai'atu, kaga wani suna Subai'atu Abubakar. No, it doesn't require ATP. Rather, it only rely on the energy created. No, it require a potential energy generated by electrochemical gradient. Please get it. It uses potential energy generated by, yes, it uses energy, of course, Gambo, Abigail Gambo. It uses energy, but which energy? Potential energy. And where do we get the potential energy? Is the energy generated by electrochemical potential. Excellent electrochemical. It relies on the energy create. No, it relies on potential energy created by electrochemical gradient. Let's get it very well. I'm going to send you that 6 minute videos. You will understand it. There are two different three videos. The second video is even differences between primary and secondary active transport. Yes, I would like you to watch that video. So I think we are done with this transport system. And then we also did concept of carbohydrate. I think the concept of carbohydrate is very clear. But do you have any questions on carbohydrates? Remember, I told you to go and get examples of each of the types of transport system. Although I tried to demonstrate that in that 6 minute videos for the primary active transport and the secondary active transport, I gave example for that. For the simple diffusion. And also I want to tell you that the secondary transport system is divided into three uniport, we have symport and antiport. So simple diffusion, glucose can use simple diffusion. So that is what I want you to understand. So by the way, so please, do you have any other question? Because I think we have shouted a lot. Can we continue or we should stop here? So most of my live video is going to be like question and answer based on what you have watched. Most of my live videos. And I'm also going to start live videos on biostatistics, the anatomy. Yeah, sorry, it is actually a network problem from my side. Okay, so Matida Job is asking question that, okay, sir, why is that most times we always insist our patients eat before taking medication? So number one, even if after you take the drugs, for the drugs to be metabolized on P450, the conjugating enzymes, They all need, they are enzymes that need energy. So to activate that enzymes, to make the system functional for the metabolism to take place, you need energy. So everything that we do, even taking the drugs to transport the drugs from one part of the body to another, up to the liver to the smooth endoplasmic reticulum, it needs energy. So energy is very important in everything that we are doing. So you need to understand that. The activation, the work of the enzymes, it needs that. So do you understand? So I'm asking question that can we continue from here or we should stop? Can we continue or we should stop? Yes, can you hear me? I said, can we continue or we should stop? Okay. So thank you very much. We are going to stop and see you next. Bye bye.